How can HSL imaging analysis investigate the influences of environmental factors on apple oxidation of various species? Every year, the USA wastes 80 billion tons of food, proving that food preservation is a critical issue that needs to be dealt with. Food waste has a major implications for food storage, transportation, and waste reduction. Tackling a problem this enormous requires a novel research method. This study uses computer vision to explore the impact of environmental factors on a seemingly simple but effective fruit, the apple. Combining an understanding of biology, chemistry, and imaging technology, the research gives an innovative and cost-effective method to observe and analyze apple decay. By the end of this experiment, we predict that if an apple oxidizes and a camera records it under constant conditions, the hue of the image of the apple's flesh, as found through imaging analysis, can predict the oxidation of the apple itself. Further, if computer vision is adopted in analyzing the hue of apple flesh, then it can improve accuracy, efficiency, robustness, and scope of the oxidation study. With the results of this study, everyday consumers will be able to pick the best fruits on their weekly outing to the grocery store. And despite this experiment focusing on apples, it can exceed the limits of just apple oxidation and possibly be used in order to benefit various food industries that are struggling with challenges in preservation, transportation, and waste reduction in our modern day. Our experimental setup consists of a temperature-controlled box in order to keep the thermal and lighting conditions consistent. The apples will be sliced into halves and placed inside the temperature control box on a labeled platter as shown below. We will have half of our apples treated with substances such as vodka or lemon juice on the top, while the other half will be the control group to compare the oxidation process. Inside the box, a white light lamp will be kept at a certain angle to avoid glare, while keeping the light constant. A camera will be attached to the scaffolding and placed above the apples. Then a blackout curtain will be placed on top of the scaffolding, covering the whole setup in order to avoid changes in lighting due to the time of day.
The camera takes pictures of the apples every 30 minutes. We then find the HSL value for the platter for calibration purposes and the HSL value for the apple half. We wrote a computer vision program that takes in all the images, retrieves and analyzes the hue values of the pixels, and performs statistical analysis. As a baseline, we also process them manually following the same procedure. After all trials, we will make graphs comparing the calibrated hue and time elapsed since the apples were first set out. Below is the process of images going through the program. First, the original image. The program will then identify sampling positions. Then it will mask out reflections and keep apple flesh, capturing hues and analyzing differences over time. The raw data includes the hue, saturation, and lightness values for both the apples and the white color reference objects, as well as the timestamps. Then we process the raw data into calibrated data by subtracting the HSL values of the apples from the color reference object, which is the platter. We also recorded the manual and automated processing times. This is the processing information from both the computer program and manual processing. As shown, the computer program processed 68,000% faster with 50,000 times more data points than manual processing.
From this data, we can see a few trends using line graphs and scatter plots. Here are 3 of them. Shown here are the changes in hue with manually inputted HSL values as well as mean value of hues over time. As the hue lowers, the graphs indicate a stronger oxidation over time, especially over the first several hours. A steeper slope indicates a faster oxidation. In the end, we conclude that the HSL model is a suitable way of measuring and analyzing apple oxidation using hue. Furthermore, using automation and computer vision makes the process a much more efficient, accurate, and magnitudinous one. Thus, our hypotheses are confirmed.
In the future, we hope to extend the scope of this study. The methods employed can be extended to analyzing more apple types and fruits. In addition, with more research, the imaging can further predict other factors, such as sweetness. For more efficiency, it is hoped that this computer vision can predict oxidation without cutting the fruit. To optimize data collection, one can look into using other color models such as LEB or YCBCR. To achieve this, the software needs to analyze millions more data points through thousands of more samples. This is the beginning to an innovative solution to food waste that can benefit numerous industries in reducing loss and providing more enjoyable foods to consumers.